Hello, everyone. My name is Deandra Spencer. I am a member of Group 4, and we will be presenting on Ebola virus outbreaks. Here, we will be discussing the introduction of the virus, epidemiology and historical outbreaks, pathogenesis and molecular mechanisms, treatment and prevention strategies, as well as a case study, and finally, summary and references. Introduction: Ebola virus is of the family Filoviridae in genus Ebola virus. The species of Ebola virus acquire their names after the region where it was originally discovered. These viruses are likely transmitted from fruit bats to primates. Here on the left we have an image of the Filoviridae virion, which has a helical nucleocapsid. We can also see glycoprotein GP, transcription factor VP30, polymerase cofactor VP35, as well as polymerase L, VP24, and matrix, which is VP40. The genome of this virus is a negative sense linear strand RNA. Because of the orientation, it is required to undergo replication to form the positive sense RNA. This will be used as a template to synthesize the negative RNA genome for each subsequent virion. Ebola virus is distinct in such a way that it synthesizes an additional glycoprotein which it secretes. This is called SGP. This helps to mediate the binding of cellular receptors. Additionally, SGP potentially plays a role in pathogenesis by binding to antibodies to suppress the host's immune system response to viral infection. Ebola virus causes a serious illness called Ebola virus disease. It often leads to symptoms like fever, bleeding, organ failure, and can eventually lead to death. The virus spreads through direct contact with infected blood or bodily fluids. Outbreaks have mostly occurred in sub-Saharan Africa. It is known for its high death rate and the need for urgent public health response.
Now that we have introduced the virus, let's take a look into its epidemiology. So, the first few cases of Ebola were seen in 1976 with two outbreaks in Africa, and the first one was near the Ebola River in South Sudan, and this is actually how scientists decided to name the virus since they didn't know a lot of information about it and it was spreading rapidly throughout the continent, causing a lot of fear and confusion. They just decided to name it Ebola virus so that people were able to identify its origin. And the second outbreak in 1976 happened in the Democratic Republic of Congo. Now, since the first few cases were seen in Africa, this virus mostly affects population from Africa, such as Guinea, Liberia, and of course DRC or the Democratic Republic of Congo, where it first originated. Now, fortunately, most of the cases recorded till today have been localized within small communities, but occasionally larger episodes occurred. And on the next slide, we're gonna see some major historical outbreaks. Now, the main source of spreading of this virus is human to human, and this means that once a person becomes infected, they are more prone to pass on the virus. Other sources of transmission include direct contact with saliva and blood, and sometimes also um, people get infected through contaminated materials, such as, for example, if you accidentally cut yourself with a needle or with a knife, and that it's a contaminated resource, then you will become infected with the virus. Also, the fact that it's a virus causes misconceptions. Many people believe that it is an airborne virus and it can spread through air, and that is incorrect. People often compare it with the flu, but that's not correct. It actually requires direct contact with an infected person or an infected container in order to contract the virus. Now, the case fatality rate of Ebola ranges from 25 to 90%, and it is really high when it compares to other viruses. And this type of complicated cases are often seen on parts of the world where medical equipment is limited or the sanitary conditions are not optimal. However, fortunately, throughout the years, early detection and appropriate medical care has contributed to an increase on survival rates, especially after the early 2000s and 2014. And you guys are going to see on the next slide why. Now that we have more background information about the Ebola virus, we are going to see some of the major historical outbreaks. And as I mentioned before, the first recorded cases were in 1976 in Sudan and also in the DRC, or the Democratic Republic of Congo. And since the virus was not yet identified at the time, scientists didn't know how to treat it or how to prevent it from spreading. And of course, this led to high fatality rates. Now, in 1995, there was another major outbreak, this time in Kikwit, which is a city located in the Democratic Republic of Congo. And scientists at the time reported 315 cases with a very high fatality rate of 81%. And unfortunately, this was because the community suffered from limited access to sanitary instruments and medical equipment. However, fortunately, since then, scientists understood the importance that sanitation and contact tracing had when it came to the prevention of more outbreaks. Now, the third historical outbreak can be seen from 2000 to 2001, and this time it happened in Uganda with over 400 reported cases. Fortunately, the fatality rates were not high because of major response effort. This time, healthcare officials began to have a more structural approach and implanted isolation guards and also educated the community on the hygiene and how it could contribute to better outcomes of the virus and a lower fatality rate at the end of the day. And finally, the deadliest outbreaks recorded to date happened in 2014, very recently to 2016. And this time it was not localized. What made this outbreak so serious is that cases not only affected African countries, but the virus also spread to parts of Europe and America. There were over 28,000 confirmed cases and almost 11,000 deaths. Now, due to the major fatality rate, these outbreaks brought a lot of international attention and collaboration to help stop the spread of the virus. And although it is really devastating that so many people were affected with the virus in 2014, this major spread led to a more research and also even the development of the vaccine that we have today.
Pathogenesis and molecular mechanisms. First, we have virus entry, where the virus uses glycoprotein GP to enter host cells. It is able to evade the host immune system by blocking interferon response by the infection of macrophages, dendritic cells, and endothelial cells by VP35, which results in vascular leakage. The Ebola virus is able to enter the body through various mechanisms, which include through the mucous membranes, breaks in the skin, or direct injection. The cell infects monocytes, macrophages, and, as said before, dendritic cells, which allows it to spread quickly through the body. This virus hijacks the human immune system or whichever organism it infects by infecting immune cells and releasing pro-inflammatory cytokines, which causes a cytokine storm. Cytokine storms are known for eliciting a overactivation or overstimulation of the immune system, which sometimes can lead to organ failure and subsequent death. The virus suppresses interferon responses, which thus weakens the body's antiviral defense. We next have viral replication. So the virus uses a whole cell machinery to replicate rapidly in the cytoplasm, as many RNA viruses do. It releases large number of new virions that infect more cells. The Ebola virus uses something called de novo replication. It is characterized by the L polymerase complex that initiates transcription by identifying the start sequence. The virus can also be transmitted sexually due to its persistence, which allows the virus to remain in semen for greater than 9 months, thus leading to resurrection of outbreaks after a while. We have the image here showing the infection of monocytes and dendritic cells. So following the pathway of the monocytes, we see suppression of interferon responses, then a result in chemokine/cytokine production, and we have a recruitment of monocytes. Following that, we have this infection affecting the liver, adrenal glands, and kidney. So in the liver, it decreases coagulation factors and it dysregulates blood pressure in the adrenal glands and kidneys. This, as we can see, has a direct effect on the blood vessels and blood, which then can lead to hemorrhagic fever, which is very consistent with what we hear of Ebola virus. Continuation of pathogenesis and molecular mechanisms. The Ebola virus is also responsible for tissue damage upon infection. Damages include the liver, the spleen, kidneys, and endothelial cells, which are those lining blood vessels, which we saw in the previous slide. This leads to vascular leakage, hemorrhaging, and multi-organ failure. Ebola has glycoprotein, GP, which is a key protein in virus entry and attachment. It also contributes to cell damage and immune evasion. The virus produces secreted glycoproteins, SGP, that bind antibodies and distract immune system. It inhibits antigen presentation and delays adaptive immune response. Some signs and symptoms of the virus include fever, aches and pains, weakness and fatigue, as well as diarrhea and vomiting. The symptoms can appear from 2 to 21 days after exposure, and symptoms generally worsen over time. Looking at this time frame of 2 to 21 days, it is evident that an individual may be infected with the virus, presenting no symptoms in the early infection and is still able to transmit it to other individuals. Additional symptoms of this virus includes unexplained hemorrhaging, redness of the eyes, skin rashes, as well as hiccups in the late stages of infection.
The treatment challenges of Ebola virus disease remain difficult. Treatment as a rapid progression and severe symptoms like the hemorrhage and organ failure are limited in healthcare and infrastructure in outbreak zones. Most of the focuses in supportive measures such as the IV fluids, oxygen, and pain management will greatly increase the survival rates of the administration early. The vaccination of the ER which is a vaccine used to modify viruses also to express Ebola's glycoproteins, training the immune system to recognize the real viruses has been a game changer, and especially during the DRC outbreaks, with over 97% protection in these trials, rapid deployment, and also the community trust were key to success. And some to reach more people with strains and future vaccines need to be more multivalent in using the mRNA platforms and also making them stable at warmer temperatures that can simply distribute. The also monoclonal antibody therapy in Mazeb, Evanga are these lab-made antibodies bind to the Ebola viruses to prevent it from infecting cells and giving the immune system time to respond, which given early that they can dramatically reduce death rates. They work by neutralizing the viruses, tagging it for immune destruction. And yes, so monoclonal antibodies should be stockpiled in high-risk areas, but with training permit partnerships, which have a plan to rotate stock and prevent expiration. So basically, for the supportive care are distilled essentials like hydration and electrolyte management, and blood pressure support is still a foundation for Ebola treatment, especially where advanced drugs aren't available as mortality drops and significantly more care is accessible early. These things are essential for supportive cares and preventions for the health and public education of Ebola transmission involves in strict infection controls, which include like PPE and isolation protocols, safe burial practices, and community outreach tailored with local customs. So the survivor educations and can be monitored. The supporting survivors means offering testing, education with mental health care, and this prevents also the post-recovery transmission, which reduces the community stigma. And the future outlook for the controlling Ebola will be requiring the medicine that it needs for a strong healthcare system, which includes a local trust and global coordination, which prepares to include vaccines, trained staff, education, and investment in infrastructure. So the monoclonal antibody therapy, which is a key breakthrough of the Ebola treatment, has most important recent advancements for the strategy of centering around lab-made proteins, which are designed to specifically target and neutralize the Ebola virus. Two of the most well-known therapies are Inmazeb and Evanga. So how does this work? Basically, the therapy that is first needed to know are the antibodies that are aiming. So the Ebola virus has a special surface called the glycoprotein that is a key to unlock and use the to enter the human cells. And think of the glycoprotein as a passcode into the virus's outer shell. Once it's inside the virus, it uses a passcode to hijack the human cells and begin replicating. So now here's where monoclonal antibodies come in. and are engineered in a lab to recognize and tightly bind these glycoproteins. And by doing so, they block the virus from binding to human cells and essentially shutting down its ability to enter and affect them. At the same time, they tag the virus, making it for destruction and localizing it by the immune system. So the result is the reduction of the amount of virus in circulating the patient's body, and with a greater chance of the body's natural immune defenses to catch up and take control. So for both therapies, the Nmazeb and the Banga, were approved by the US FDA. The Nmazeb has three antibody cocktails, which are targets different parts of the Ebola glycoproteins, which has a stronger and more comprehensive neutralization. While the Ebanga therapy has the other hand monoclonal antibody, which is simpler to produce and store while provides targeted effective protection. These therapies were also investigated and tested during 2018 and 2020 Ebola outbreak, and that was from the Democratic Republic of the Congo, which it also improved dramatically the survival rates, especially when they were giving early these course of infection. So the monoclonal antibodies can be stockpiled in high-risk zones. Yes, so for example, the Ebola outbreaks, having antibody treatments on hand and ready to deploy means that we can immediately begin treating patients before the disease progresses. High-risk regions like the West Central Africa should be regional stockpiles and supported by international health organizations like the WHO, and MSF, training local and health workers to administer these treatments and recognize early symptoms is just as important as drugs themselves. And these challenges of monoclonal therapies are costly and need cold storage, which are administered intravenously and must require planning, resources, and partnerships. So the major leap forward were not one but done solution, and they must be part of the larger response strategy for them to detect the public for early detection and give the public the free education that they need. And to conclude, the antibodies of the monoclonal therapy, the transformational advance in treatments for the Ebola were by directly attacking the virus and helping the immune system fight back. And these therapies reduce death rates and offer real hope during the outbreaks. So by combining these early detection supportive cares and public education, and they have potential thousands of saving lives. Not only they're already available locally for these therapies, but they're also backed by global commitment.
This case study focuses on the 2014 West Africa Ebola outbreak, the largest and deadliest in history. It began quietly in a small village in Guinea where a young boy fell ill with an unknown sickness. His family did what any family would do. They cared for him with their own hands, not knowing that this act of love would spark a chain of events that would change the region forever. From that single village, the virus spread rapidly, crossing into Liberia and Sierra Leone. These countries were already struggling with limited healthcare resources, and the outbreak quickly overwhelmed hospitals, clinics, and communities. Nurses, doctors, and volunteers worked tirelessly, often without enough protective equipment, and many became infected themselves. Some lost their lives trying to save others. The way Ebola spread made it even harder to contain. It passed through direct contact with bodily fluids, and in tight knit communities, people cared for their sick loved ones at home. Traditional burial practices, which involved washing and touching the bodies of the deceased, also played a heartbreaking role in its spread. By the time the international community fully mobilized,
with organizations like the WHO, CDC, and Doctors Without Borders stepping in. The crisis had already taken hold. Entire villages were devastated. Families were torn apart. In total, over 28,600 people were infected, and more than 11,300 died. Yet among the tragedy, there was resilience. The outbreak forced the world to rethink how we respond to global health emergencies. It led to stronger international collaborations, better preparedness protocols, and most importantly, the fast tracking of the first effective Ebola vaccine. That vaccine has since been used to help stop future outbreaks before they spiral out of control. The 2014 outbreak left deep scars, but it also sparked hope. Hope that with faster action, better tools, and greater compassion, we can do better next time.
As the presentation comes to an end, summary and key takeaways include Ebola virus remains a persistent global health threat, with outbreaks triggered by zoonotic transmission from animals to humans, human behaviors such as traditional burial practices, and fragile healthcare infrastructure in affected regions. Some major outbreaks like the 2014 to 2016 West African epidemic and the 2018 to 2020 DRC outbreaks has revealed the devastating impacts of delayed detection and the poor health system capacity. With understanding the virus's molecular biology, the treatments have advanced. Ebola uses proteins like VP35 and VP24 to block the host interferon response, weakening the early immune detection. The glycoprotein GP plays a dual role in allowing the virus to enter host cells as well as damaging immune cells like dendritic cells, macrophages, and giving Ebola sort of a head start in infection and immune evasion. Long-term immunity and viral persistence pose continued challenges. Some Ebola virus survivors have the virus in immune privileged sites like the eyes or reproductive organs, with rare but documented cases of reactivation or sexual transmission months after recovery. A resilient global response requires a multidisciplinary approach. Scientific breakthroughs must be matched with strong healthcare systems, early detection networks, and culturally sensitive outreach. New insights into molecular replication mechanisms present new targets for antiviral drugs. Immunity with vaccination is vital to minimize outbreaks and contain the spread. Finally, future research should focus on post-exposure prophylaxis and long-term immunity. Here we have a list of references used to put together the information found in this presentation, which includes the de novo replication, emerging zoonotic disease, the replication of the virus, hemorrhagic fever, etc. Finally, on behalf of my group members and I, we would like to thank you for listening to our presentation, and we hope you learned something new.